Hi, myself Gautam Kumar TM from Shivanteshwar College of Engineering, Bangalore. Today I am going to explain about nucleic acids. Let, First, let us look into the contents. Today I will be going to be explaining about what are nucleic acids, types of nucleic acids that are DNA and RNA, and applications of nucleic acids. First, DNA vaccination for rabies, RNA vaccination for COVID-19, and DNA fingerprinting. All these topics will be covered in this video. First, let us look into the introduction part. Nucleic acids are naturally occurring chemical compounds that serve as a primary information carrying molecules in cells. They play a special important role in direct potential synthesis. The two main classes of nucleic acids are DNA and RNA. Now, let's move into the main topic. What are nucleic acids? Nucleic acids are biopolymers and micromolecules, essential to all known forms of life. They compose of three main compounds, which which are made up of three compounds, that is 5 carbon sugar and phosphate groups and nitrogenous base. The two main classes of nucleic acids are DNA and RNA. Nucleic acids are a naturally occurring chemical compounds that serve as a primary information carrying molecules in cells. They are made up of genetic materials. Nucleic acids are found in all form of living things, whether they create or encode or they store information in every cell of every life form on earth. It turns their function into transmit and express the information inside and outside the cell molecules into the interior organization of cell and ultimately to the next generation for each living organism. The encoded information is contained and converted in the nucleic acid sequence, which provides a ladder step ordering of nucleic within the molecules of RNA and DNA. They play a special important role in direct protein, protein synthesis. Now we move on to the DNA vaccination for rabies. DNA vaccination, rabies vaccination. It's a simplest embodiment. It is a euteric expression of vector containing the rabies virus glycoprotein genes. Under a suitable promoter, DNA vaccine on based of electro-targeted glycoprotein of rabies virus. It is impartial potential of 60% against the 20 LD50 of challenge virus standards CVS. Rabies, progressive fatal empathize. It is caused by the rabies virus of genes Lavet virus. The major of rabies cases reported from developed countries involve wild animals like raccoons, bats, fox, etc. DNA vaccination being free from foreign proteins may not cause various side reactions. It may be obvious for conversion of vaccination vaccines. Now let's see the ways, 8 steps of making DNA vaccination for rabies. DNA vaccines. First, RNA is extracted, then RNA is converted to DNA representation, then we generate the sequence from DNA and we take the antigenic proteins are amplified from the DNA that we generated from before. Next, we amplify the frequency of the target genes is cloned in various expression vectors. Then it is carried through a large quantity in bacteria. Then it is then plasma is purified and injected to the host as vaccine. Now let's move on to the RNA vaccine for COVID-19. This is a type of vaccine gives to your cell instruction for how to make S proteins found in on the surface of COVID-19 virus. After vaccination, your muscle cells being makes S potential shapes displaying on the cell surface. This cause your body to create antibodies, improves the stability of mRNA to increase the fertility to manufacture RNA based products to decrease RNA associated inflammatory response have to lead a significant advances in development of mRNA vaccination. And there are several reasons that mRNA platform technology as emergency as referral vaccination technology. Among this rapid speed, the mRNA candidate vaccines can be constructed and manufactured and is need necessary to rapid development vaccines against a newly emerging viruses. The potential of mRNA vaccine as a technology to rapidly growing response public emergencies of influenza diseases. In addition to the application of the vaccine from the additional influenza diseases, we underscore integral of other RNA vaccines. These are the six steps that is needed to develop the RNA vaccination. Now let us move on to the next one, DNA fingerprinting. DNA fingerprinting is a laboratory technique used to determine the formidable identity of a person on based on nucleic sequence of certain regions of human DNA. That is a unique individuals. DNA is present in nearly every cell of our body. It may contain flakes, skin, drop of blood, hair, saliva. All of these contain DNA that can be used to identify us. In fact, the study of forensic, the commonly used by police department around the world, frequent related to some small bits of DNA to link the criminals to the crime they committed. This is a fantastic science is offered to popular on television so as simply exact for method to find the potential being him or her to Judy. In truth, however, tracing of DNA fingerprinting determination of likelihood of match between a suspect and a crime scene is completely progress related upon a probability to greater extent than more the people realize. Government administrate the DNA database such as combination of DNA index system C CODIS to help the speed process but also bring the lightly complex ethical issue involving the right and of victim and suspect alike. Thus, the understanding of the way in which the DNA evidence is obtained and analyzed, and however this evidence is used within the legal system. This is a critical appearance of true ethical and legal impact of forensic science. These are these are some points of DNA fingerprinting. At last conclusion, nucleic acids are essential biomolecules that store and transmit the generating information in living organisms. DNA serves as a blueprint of life, which while RNA plays a critical role in protein synthesis. Understanding the nucleic acid is a fundamental to our knowledge of genes and other significant implements of various fields, including biotechnology and medicine.